Hello friends, welcome back to my channel, and today in this video we are going to study gene-gene interactions. Now, when there is no interaction, in that case, the ratio we obtain in a dihybrid cross is 9 is to 3 is to 3 is to 1. This we already learned that this ratio is when the two genes are not linked, the two genes are not interacting with each other, in which this 9 is for the dominant phenotype. This 1 is for the recessive phenotype. And this middle 3 and 3, these two are for the hybrid of dominant recessive character. That means one character should be dominant, the other should be the recessive one. So here for 9, the genotype should be A dash B dash. Why A dash B dash? Because if in this dash, if I write capital A, capital B, or if I write capital A, capital B, small b, in both the cases, this dominant one will express. Doesn't matter in dash, we write it capital A or capital B, small b. In either cases, the dominant phenotype will be visible because one dominant allele is present. So that's why it is written capital A, capital B, because this 9 is for dominant phenotype. And when we will get dominant phenotype? When at least one allele dominant is present. Because with this dominant allele, if another dominant allele is present, then also the phenotype will be the same. If one recessive is also present, in that condition also the phenotype will be the same because the dominant allele will express itself. So therefore, the 9 ratio is for A dash B dash, that means for the dominant phenotype when both the characters are dominant. 1 is for small a, small b, that is the recessive. Both the character are in recessive state. The middle, both the three, these are for one character should be dominant. Here you see capital B is written and here capital A is written. That means here A is recessive, B is dominant, and in this one A is dominant and B is recessive. So the 9 ratio, remember it is for both the character dominant. For 1, both the character recessive. For middle 3, one should be dominant, the other character should be recessive, and for the next 3, vice versa. This is the phenotypic ratio of dihybrid cross, that is 9 is to 3 is to 3 is to 1, when both the characters or both the genes are not in interaction with each other and they are also not linked with each other. Now, first type of gene interaction, which we call it as a collaborator gene interaction. What do we mean by the word collaborator? That means when two different genes that are present at different locus, they are contributing together for a single phenotype. For example, here if you see, when one allele is dominant for one character A, it is giving red color. The second gene is in recessive state. Here, the second one is in dominant state and the first A gene, it is in recessive condition. Then the phenotype we are getting is green. When both are in dominant condition, in that situation we are getting brown colored. That means here gene A and gene B both are contributing together for a single phenotype and hence we call it as a collaborator gene interaction because both the genes A and B are collaborating together for a particular phenotype. When one character or one gene is in recessive state. For example, in this capital A, small b, B is totally in recessive condition. So who is expressing? Only capital A is getting expressed and the color we are getting is red. Similarly, when B is dominant, only B is getting expressed because A is totally recessive. Here we are getting the color green. But when both are dominant, then we are getting the color brown because both are collaborating together for a particular phenotype. So this is one type of gene interaction, that is collaborator gene interaction. Earlier we studied allelic interaction. Don't get confused between allelic interaction and the gene interaction. Those who have not seen that video, I would request you to once see that video to understand the difference between allelic interaction and the gene interaction. Now, the second type of gene interaction is synergistic gene interaction. Synergistic gene interaction means when double mutants shows better phenotype than wild type. For example, here if you see again, gene for one character is dominant, the second one is totally recessive. So here the phenotype we are getting may be the inferior phenotype. In the reverse condition, when B is dominant and A is recessive, Again, we are getting the inferior phenotype. But when both the genes are in recessive state, then we are getting the better phenotype. So such type of gene interaction where we get better phenotype when both are in recessive state, it is known as synergistic gene interaction. Now, polygenic gene interaction. Polygenic gene interaction means when two or more genes contribute to same phenotype in additive fashion and give average traits. What do we mean by this? For example, this is parent 1 and this is parent 2. Parent 1, a pure homozygous dominant. Parent 2 is a pure homozygous recessive. So when capital A, capital B giving violet color is crossed with small a, small b giving white phenotype are crossed. In F1 generation, we are getting capital A, capital B, small b, and the color we are getting here is red. When the members of F1 generations are crossed to get the F2 generation, there we see that we are getting a range of colored phenotype from violet to white. When we are getting these colors, when capital A, capital B are present together, it is giving violet color. See, all the four alleles are in dominant condition. When the genotype is capital A, capital B, small b, or capital A, small a, capital B, then we are getting dark red color. Here, if you see, three alleles are in dominant condition. When the situation is capital A, capital B, capital A, small b, or small a, capital B, or capital A, capital B, small b, that means two alleles, two alleles. In this situation also two alleles. So when two alleles are dominant, we are getting red color. Clear? For pink, it is capital A, small b, or small a, capital B, small b. That means only one allele is dominant. Then we are getting pink color. When zero allele dominant or when no alleles are dominant, in that condition, we are getting white color. So depending upon the number of dominant alleles, we are getting a range of phenotypes. So this is known as polygenic gene interaction. How we can calculate the probability for these genotypes? Already told you, whenever dihybrid, trihybrid, such situations are there, always considered as a monohybrid. Single character. So this is capital A separately. This is capital B separately. If you remember, in a monotype, capital A, when what is the probability? 1 by 4. Why? Because in monohybrid cross, the ratio is 1:2:1. 1 for homozygous dominant, 1 for homozygous recessive, 2 for heterozygous. Right? So capital A, 1 by 4. Similarly, capital B will also give 1 by 4. So 1 by 4 into 1 by 4, that is 1 by 16. How you will calculate this? For capital A, 1 by 4 into capital B, small b, that is 2 by 4. This situation or in this, whenever in probability we use or, we have to add it. Whenever in probability we use the word and, we have to multiply it. So here both the characters together. So capital A and capital B, small b. So both the characters are together. So here we are multiplying. In this genotype also we will get dark red, or in this genotype where capital A, small a, capital B. So capital A, small a, that is a heterozygous situation, 2 by 4 into BB, that means 1 by 4. So the final probability will be 4 by 16. Clear? Understood how we calculate probability? This we are learning from last few videos. Okay. So now it's your homework to finish the probability for red, pink and white. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.